So let us start with a very fundamental question. Why should we care about the NGS data quality? So what is the need to assess the quality of the data we are getting out of the sequences? So for that, imagine you have invested a lot of money in a sequencing experiment, and you are preparing, you have prepared the samples and you waited for weeks to get the results. And finally, you have received a file with millions of reads. But what if most of those reads contain errors? Or what if your sequencer had a technical issue during the run? Or what if the quality declined halfway through? So if you skip the quality assessment and proceed directly to the downstream processing, that is the assembling part, you'll lead to a you lead to generate a waste product out of it. So this brings us to the classic scenario of garbage in and garbage out. So if you use the bad quality reads, that will give you misassemblies and false biological conclusions. And also that processing of bad data will waste your computational resources and money. And even if you get something out of it, that results will be not publishable because they are not reproducible. They are irreproducible. So quality control is your first line of defense if you are if you are going to work with them. So it's like inspecting the foundation before building a house. Now, this is for your knowledge. So where do we get these data from? So most of the researchers, what they do, they sequence their data, and before publishing any analysis or any results out of that, they submit it to the SRA database, which stands for the Sequence Read Archive. So you have come across some gene announcement or any sequencing related articles where you find that data availability section. There they refer to some data. That data is the raw sequencing data, and that data is publicly accessible. So that data is stored at this database, so anybody can access it and work out the same protocol what you have followed to reproduce your results, to validate that you have done something real. Then what is Sequence Read Archive? So it is a repository, the world's largest public repository for raw data sequencing. It is managed by National Center for Biotechnology and Information, NCBI, and you will find every kind of sequencing data there, whether it is sequenced using Illumina sequences or PegBio or Nanopore and etc. Uh, what data you could find there? So that would be FASTQ files, the raw sequences, the metadata, like where these samples are collected from, and the experimental protocols you followed during preparation of the samples or the preparation of the libraries, and then sequencing platforms, of course, what instruments you have used to sequence them. Then how do you access this data? So there are two ways. You can either use command line tools like SRA toolkit, which uses fastq dump or fasterq dump commands to download it, or you can either go through uh, cloud-based accesses like AWS or GCP, or you can use their the web server of NCBI SRA database simply by searching SRA NCBI database. You can find that. And how can you like how the data is structured there? So you can find the data by using the bioproject accession number or the biosample accession number or the SRA run ID. Okay, so yeah, that's all about the SRA. So once you get the sequences, what you'll do next? So we are going to assess the quality. So the next thing is let's let us talk about what is quality in context to next generation sequencing. So uh, every base, every nucleotide base in your sequencing read has an associated quality score, something you call Fred quality score or Q score. So this Fred score represents the probability that a base is called incorrectly, meaning the machine identified it incorrectly. So it is calculated as uh, negative of 10, means minus 10 log base 10 multiplied by the probability of getting the base incorrect. So we get this quality scores. So what does this course tell about the sequence? So if you get a quality score of 20, uh, which this means that there is 1 in 100 chance that the base is wrong. Or if you get Q30, this means there is 1 in 1,000. And if there is a Q40, this means there is 1 in 10,000 chance that the base is incorrect. So you can see this pattern. The higher the uh, Fred quality score, the better is your sequence reads. Okay. So a quality score lower than 20 is questionable, that can be unreliable, or a quality score between 20 to 30 is a good, it's considered good and acceptable, and a quality score greater than 30 is the gold standard and very excellent for downstream processing. But if we look at it from a perspective of for a single read sequence, the quality isn't uniform across the read. Typically the quality is higher at the beginning, that is the 5' end, and it uh, degrades as we move towards the 3' end of the sequence. So what could be the reasons? So, the reason might be sorry reason might be photo bleaching. That is the fluorescent dyes which are used in the sequences may fade off over time, or there is accumulation of many errors that gets compounded and becomes a bigger error as we go deeper into sequencing. So understanding this pattern is crucial for us to decide what part we need to remove. Basically, what part to trim off. Okay. So now again, a bigger question is, how do we get this quality score? So a dedicated tool is there, which we call Fast QC. It is one of the most popular tool which is used for assessing the NGS quality, NGS data quality. So FastQC is a Java program that analyzes the FastQ files and generates an HTML report with multiple quality metrics. Think of it as a comprehensive health checkup for your sequencing data. So what matrices do we have here? So these we will again discuss when we do the hands-on. So first is the per base quality, which tracks the quality scores across the read length, revealing positional degradation patterns. Second is per sequence GC. So for each sequence, what is the GC content? It shows the GC content distribution, which should match your organism's expected profile. So there is this thing that every organism has a range of specific GC content. Uh, so whenever we uh, check the GC, it should fall in that respective GC range. If it is falling above or below that, that could be a indication of contamination. Third is N content. So N is basically the character which is assigned to the position in a sequence when the sequencer is not confident enough to assign which base is there. So to compensate that, it puts an N. So N could be anything, any of the four bases. Then fourth is duplication levels, which measures the read redundancy. So how many copies of a read, identical copies are there, which could indicate that PCR biases or any issues or mishappening which occurred during the natural library preparation. Then fifth is adapter content. So the adapters, as you know, adapters are sequences which are used during sequencing to attach the primers to the flow cell. So there could be some remaining adapter content. So this also, the FastQC also gives you the uh, sequences. It detects that the adapter content. And sixth is the sequence length, so which confirms the expected length, read length distribution across your data set. So it estimates and that how much sequence should be there, sequence length should be there, which it calculates using coverage. We will discuss coverage in later sessions. So yeah, let's move forward. So FastQC is a powerful tool, but when you are processing dozens or hundreds of samples, reading individual HTML reports for every sample, that becomes a tedious task. So here comes the multi QC. So it it's a tool that will aggregate all these results, all the reports out of the fast QC analysis, and brings you a single comprehensive HTML report which can be used to compare different matrices in between the samples, and it can let you detect any outliers if there are any uh, uh, samples which fall very uh, away from the expected ranges of different matrices. Uh, yeah, and it gives you a single and interactive dashboard. So it also provides heat maps for quality patterns, and it can also be used to detect batch effects, like uh, do certain samples cluster together, or trends like is quality degrading across the run, so on. uh, yeah, multi-QC reads the output of fast QC and presents them in a way that makes it easy to spot patterns you might miss during um, uh, basically if you check uh, single reports, you might miss individual reports for every sample, you might miss something. So multi-QC provides very uh, what do you call it? Uh, okay, let it be. So next, what common issues do we face during quality control? So yeah, high-end content. So sequencers couldn't confidently call bases at specific positions. The same thing I've discussed. It often indicates optical issues or chemistry problems in the sequencing chemistry. And what actions you could take? So you can investigate the sequencer logs, or you can consider resequencing, which might be a very cost effect, very costly thing. Again, you will uh, be in a loss. And then we have multimodal GC distribution. You can find multiple peaks in GC content, which again, same thing I've discussed, that there can be contamination in your sequence. So what you can do is you can blast suspicious sequences against the NR database, the NCBI database of FASTA file or FASTA sequences, which is there. Uh, and there could be high duplication rates, which indicate the PCR over-amplification and library complexity issues. Then quality score cliff. So there can be a dramatic quality drop, which suggests the instrumental malfunction, and persistently low base quality. So over poor quality across overall, poor quality across reads requires aggressive trimming. Then, which is our next step? We are going to trim off the low quality bases using a trimming tool. So, by analyzing multi-QC report and fast QC report, we get an idea to what uh, parts to remove from your sequences. So, once we get that patterns and understand what to remove, we move towards the trimming part, and trimming removes the low quality bases from end of the reads. So as you know, the quality degrades when we move towards the 3' end. So we remove the ends, 3' ends most, but it is not always necessary, because if your data is of high quality, you will get to know by doing the fast QC. You might not need to trim at all. So yeah, Again, if your quality score is greater than 25, the Fred score, then you don't need, you don't need, you might not need trimming. And these are general guidelines. And if your quality score is in between 20 to 25, moderate trimming can be done. And if your quality is less than 20, then aggressive trimming is required, or might there might be a need to discard some reads entirely. So what tools we use for trimming? So there are three very popular tools. First is Genomics, which is a Java-based, highly configurable tool that takes paired end data. Uh, I suppose you guys know what is a paired end data in context to FastQC uh, in FastQ files. Uh, can you guys say yes or no in the chat box? You know about paired end data. What is paired end sequences? Okay, you don't know what is paired end. Okay, so basically, okay, I'm explaining in briefly. So basically, paired end sequencing is when a sequences a read. First thing is we take the DNA and different fragments are generated. So when the fragment is sequenced, it is sequenced from both end, the 5 prime end as well as the 3 prime end. So there will be one copy of read of that fragment from its 5 prime end towards the 3 prime end, and the another copy from the 3 prime end to 5 prime end. So there will be lesser chances that some regions we might miss can come into our fast QC file. Do you get what I'm trying to convey?
Sir, can you please repeat?
Yeah. So when we sequence a fragment of DNA, uh, it gets sequenced two times. Once from its 5 prime end to 3 prime end, and again from its 3 prime end to 5 prime end. So we will have two copies of a fragment of DNA. So these two copies are called paired ends. So for 5 prime end to 3 prime end data, there will be a separate FASTQ file, and for the 3 prime end to 5 prime end sequence, there will be a separate FASTQ file. So we will have two FASTQ files for the same set of data. Do you get it? So these are pairs.
Okay, sir.
Uh, okay, so I suppose you guys are getting it. So we'll move forward. Yeah, so once we have trimmed off the sequences, we will recheck the quality of the data. Again, we will repeat the fast QC and multi QC, uh, and again check for the quality. And even if we have done the trimming, there might be some sequences, some part of sequences left with lower quality. So if there is some low quality part, we can again trim it and again recheck for the fast QC and multi QC. Okay, so this is the whole workflow for quality assessment. First, you generate a fast QC out of the raw sequencing data. Then, if there are multiple samples, you can create a multi-QC report. Then you analyze it, you interpret some results out of it, that this part is of low quality and we can remove it, or whether uh, uh, no trimming is needed at all. That decision we take at this point, and then we set, if trimming is needed, we set parameters, what to be trimmed off, and then we execute the trimming, and finally we'll get a better high quality read sequences. So that's all for the theoretical part. uh, if you guys have any questions, you can ask. Then we'll take a 5 minutes break and move on to the hands-on part.